You codons are always written in triplets. There is no gap in DNA, but for easy understanding I have put some gap there. So GCC, TAT, TTG, this is the information on the DNA. Now if the information on DNA is this, then what should be the mRNA formed? CGG because complementary, TAT complementary will be AUA, TTG complementary will be AAC. Now let's understand from the NCERT. If CGG is there, if CCG is there, it will cause, where is it gone? CCG is there, it will cause, CGG is there, it will cause the formation of arginine. Which amino acid? Arginine. If AUA is there, it will cause AUA. If AUA is there, it will cause the formation of isoleucine. And if AAC is there, it will cause the formation of asparagine. But now let's understand. Imagine that there is an inversion. Now what inversion is there? Inversion is taking place. Inversion is what? GCC, TAT, TTG. Now this whole thing undergoes inversion. So if it undergoes inversion, what will happen? GTT will come first, TAT will remain as such, and this will go at the back. Now if it goes at the back, what will happen? CCG. Now this has become GTT. So because the whole thing has undergone inversion like this, now this will cause formation of a different type of RNA or mRNA. Remember, this is the information which I have mentioned here. This is the information of the mRNA. So once the mRNA information changes, what will happen now? This will be complementary. This is the complementary mRNA now. This is the complementary mRNA now. This is the complementary mRNA now. So once the mRNA codons have changed, they will not code for same amino acid. The amino acids are going to be changed. So why these amino acids have changed? I think the reason has become clear to you, because codon information has changed. So once a DNA undergoes even a slight change in the information, it will cause mutation. So inversion has caused different type of amino acids to be formed. In the same manner, it is possible to have transition. Now what is transition? Transition means when a purine is replaced by, transition. Sorry for that. What is transition means when a purine is replaced by a purine and a pyrimidine is replaced by a pyrimidine. Or what is transversion? A purine replaced by a pyrimidine and a pyrimidine replaced by a purine. So that means similar nitrogen bases, because we know A and G are purines and C and T are pyrimidines. So when A is replaced by G, it is called as transition, or when C is replaced by T, it is called as transition, or opposite also. But if A is replaced by T, or otherwise you can say if a C is replaced by G, then you will call them as transversion. So let's understand this concept, that what change will happen. Imagine the sequence of the DNA is same again, GCC, TAT, TTG. So the mRNA form should have been this. Arginine should have been come, leucine should have come, asparagine should have come, because this would be the reading on the mRNA because of this. But if transition occur, now what is the transition? G is replaced by A purine replaced by purine. Now this is the new DNA. Now this new DNA will not cause the formation of same mRNA. Now the mRNA will change. Once the mRNA change, amino acid formed also would be changed. In the same manner, transversion is possible. What is transversion? A purine replaced by a pyrimidine, or pyrimidine replaced by a purine. So if G is replaced with C, G is a purine and C is a pyrimidine. So this transversion will cause change in the DNA. Once this single change, just imagine a single change has occurred, this single change will cause a different type of mRNA code, and once the mRNA code will change, this will cause the change in the amino acid. So instead of arginine, now glycine will come. That's why a single base change can bring changes. So now remember one more thing, a single base change means point mutation. What do you call them as? Point mutation, because only one base has undergone a change. Now in the same manner, insertions are also possible, deletions are also possible. So what are insertions and deletions? In the last sheet we had understood that it can be number change, sequence change and type change, and frameshift mutation. So inversion we have learned, substitution we have done, now we are doing the frameshift mutation. Frameshift mutation is because of insertion, so addition of something extra, or deletion would be called as the frameshift mutation. Now imagine ACC, TAT and TTG is the information on the DNA, and you insert an additional A here. Once you insert additional A here, reading will change. Now the reading will be AAC, then CTA. So reading is going to change from one step. From which step? From the step from where you have added the new information. From that point onwards, the reading is going to change. So once the reading change, in the same manner, the type of amino acids, all. Now here you can say only one amino acid was changed. Here all of them would be changed, because from the point of insertion, the whole reading is going to change, whole reading of DNA. And if you remove one nitrogen base or nucleotide from this whole sequence, again same thing, from the point of deletion onwards, the whole DNA reading is going to change. And once the reading is going to change, the amino acids formed also would be changed. We will continue this topic in the next.